The nervous system has two categories. We have the central nervous system, and then we have the peripheral nervous system. The central nervous system consists of the brain and spinal cord. And the peripheral nervous system, it includes the cranial and spinal nerves. And as you might know, cranial nerves are the nerves that take signal from the brain to the regions of the face and the head. So they take information to and from the brain. Whereas spinal nerves, they take nerve signal from the spinal cord, or sometimes they take the signal to the spinal cord. The other way of looking at the peripheral nervous system is as a two-category system, the somatic nervous system and the autonomic nervous system. The somatic nervous system is the part of the peripheral nervous system that provides nerve supply to skeletal muscles. It innervates the skeletal muscles, and most of them, as you know, the somatic nervous system manages the voluntary movements of the body. And the somatic nervous system has both sensory and motor systems. That means sometimes it takes information to the central nervous system, and sometimes it brings information from the central nervous system. On the other hand, the autonomic nervous system, it innervates the cardiac and smooth muscles and glands, which tend to be involuntary. And unlike the somatic nervous system, it has only motor systems only. It takes information away from the central nervous system instead of taking it toward the central nervous system. Autonomic nervous system, or the involuntary portion of the peripheral nervous system, has two categories. Those are the sympathetic division and the parasympathetic division, or sometimes called the sympathetic nervous system. This is the part of the nervous system that controls the fight or flight responses when we are faced with moments that tend to increase our heart rate, blood pressure, and things like that. And the main neurotransmitter in this nervous system is the norepinephrine. On the other hand, the parasympathetic nervous system focuses on rest and digest responses. This is the kind of nervous system that's activated when we are in a calm and quiet state. That's why it's called the housekeeper division, because this is the time when you do all the regular stuff, like when you are eating, when you are relaxing. The parasympathetic division is being activated. And the main neurotransmitter that's being activated in the parasympathetic nervous system is the acetylcholine. So this is all regarding how the nervous system is categorized. The next thing is, what are neurons? Like, these are the categories of nervous system, but what are the building elements of the nervous system in general? And as you know, neurons are the functional units of the nervous system. And neurons have a cell body. Cell body is the one, is that round structure that you can find sometimes at one end of the neuron, or sometimes in the middle of the neuron. And the cell body is the part of the neuron that contains the nucleus and the other organelles. And then we have the dendrites that receive signals either from sensor receptors or from other neurons. These tend to be these are like the branch-like structures that you find closer to the cell body. And then we have the axon, which is the long structure that propagates the nerve signal in the form of action potential. And two things you need to know about axon is sometimes they might be covered by melanges, which is made of lipids, and it covers around the axon. It's like a covering for a wire. The axon is the wire, and when you try to cover, when you try to insulate it with some kind of plastic, that's like the melanges. And melanges is interrupted by the nodes of run V. These are like, uh, let's say if we keep the wire analogy, so nodes of run V is, let's say, if you cut in the middle of the wire at a regular interval, if you expose the wire, that section is called node of run V. This is the unmyelinated section of the axon. Now, we know what neurons contain and how many types of neurons are we familiar with. We have three different types of neurons. The first one are sensory neurons. These are neurons that take signal to the central nervous system. And we have interneurons that are entirely found in the central nervous system only. Sensory neurons, they are extended from between like the central nervous system and the rest of the body, but internal neurons, they are found within the central nervous system. And motor neurons, they take signal from the central nervous system to the effectors. In this class, when we say effectors, where we mean the muscles and the glands that take signal from the motor neurons. So these are the types of neurons. And then we have the nerve impulse. When we say a nerve impulse, it is this electrical signal that travels uh, through the neurons because of an action potential. And this action potential, or the reason why nerve impulse happens in the first place, is caused by concentration gradients. And when we talk about concentration gradients, the first thing that comes to our mind is the sodium potassium pump. So I'm not going to ask you to understand the details, but there are certain things that you need to commit to your memory because they tend to come again and again, and it is better to have at least the main idea of how this sodium potassium pumps and how it drives the action potential. So when you think of depolarization, this is something that happens when sodium rushes into a neuron. Uh, by default, almost most of the cells tend to be negative in the inside and positive on the outside. That means the relative electric gradient is the cells tend to be negative. So since it is a negative state, when you say depolarization, you are trying to make it less negative. And it looks, it's actually pretty obvious because sodium has a positive ion, and when sodium rushes into the cell, it's going to make this inner side of the cell less negative. That's why it's called depolarization. And instead of pushing it to the polar, let's say this north pole and south pole, and as I told you, the cells tend to be negative. So let's say they are on the southern end or the south pole. So when sodium rushes in, it's going to bring it up. It's going to try to make it a little positive. So it's going to bring it back to the equator, relative speaking. So depolarization is something that happens when sodium rushes into the neuron. Repolarization is when the potassium leaves the cell. It's like now the electric gradient is going to go back to where it was before, like it's going back to the south pole, because if the positive ion is going out, that means the cell is going to regain its former electrical gradient. And there are times where hyperpolarization might occur because of the sodium potassium pump or other ion channels. And one example is what happens to the cell if, let's say, chlorine ions rush into the cell. And as you know, chlorine has a negative ion, and if a negative ion rushes into the cell, it's going to make the cell more negative. And that's just going to become hyperpolar. This is all regarding nerve impulse. And then axons. As we have said it earlier, axons could be myelinated or they can be unmyelinated. If they are not myelinated, that means if they don't have any myelin sheaths, the signal is going to be a little slower. It's not going to travel that fast. And since it is unmyelinated, the signal is going to travel like a ripple effect. That means when there is a sodium potassium pump, the action potential in one part of the neuron is going to affect the action potential on the next sodium potassium pump. So it's like the signal is traveling on a contact basis, one touching the other, like a ripple effect. And the speed of nerve signal in unmyelinated axon is 1 meter per second. And if it is myelinated, it's going to go faster. It's like a wire that is properly insulated. It could go up to 100 meters per second. So in myelinated axons, the way the nerve signal or the action potential travels is different from unmyelinated axons. The reason is because of the myelin sheath. The action potential cannot just travel as a ripple effect or as cannot travel in contact basis. So in saltatory conduction, it's the signal, the action potential is going to jump from one node of Ranvier to the other. So in myelinated axons, this is how the action potential travels. Now, what's action potential in the first place? Action potential is the nerve impulse that's generated because of the concentration gradient. And it happens in an all or none phenomenon. Either you have action potential or you don't have action potential. So if the concentration gradient, if the stimulus is strong enough and the concentration gradient is disturbed, probably this might create an action potential. It creates an action potential, it's going to travel along the axon. And if the stimulus is not strong enough, that means there will not be an action potential because it doesn't hit the absolute threshold. So you will not have an action potential. Action potential can be affected by the time and space of other action potentials. Let's say there's one action potential we refer as A. And if there are other action potentials like in terms of physical and how could I say, like time-based interval. If there are other action potentials close by in terms of time or space, this might affect other action potentials. Let's talk about the time. If two action potentials happen at the same time, one of the action potentials might actually either increase or decrease. That means either it might amplify or it might reduce the action potential of the other. I'm going to create another analogy here. Let's say one action potential has a magnitude of -1 megavolt or millivolt, sorry, -1 millivolt. If the other has also -1, -1 plus -1 is going to be -2. So it's going to make it polarized. It's going to push it down. But if it was one of them was -1 millivolt and the other one was +1 millivolt, you will not have an action potential because it's going to cancel out. The same thing in terms of space. If two action potentials happen close by in terms of space, they might add up. That means they might amplify and sometimes they might cancel out to each other. So action potentials that might amplify or reduce the action potentials of the other signals based on time, the situation is called temporal summation. And the action potential that might amplify or reduce because of space, that is called spatial summation. Time and space, temporal and subspatial summation. Another important phrase I want you to remember is refractory period. Refractory period is once you have an action potential uh, for, a, for a certain interval of time, there will not be any other action potential, no matter how much the magnitude of the stimulus is high. This is called the refractory period. No signal immediately after an action potential, because it is as if the neuron needs to rest every time it goes through one action potential. It needs rest. And one thing you need to remember: action potential travels in one direction. It is a one-way street. You will not have action potentials that go both ways. Synapse. Synapse is the space between two neurons in the central nervous system, mostly. It's between two neurons. And if we are talking about the space between a neuron and a muscle, that space is called neuromuscular junction. So neuromuscular junction is a special form of synapse. In the synapse, a nerve signal stops at the axon terminal of the presynaptic neuron. A nerve signal or the action potential stops there. And instead of the nerve signal, because the nerve signal cannot go through the synapse, it stops as soon as the axon terminal ends. So instead of this nerve signal or electric signal, what's going to happen is a chemical signal is generated, like that's equivalent in terms of strength, equivalent to the nerve signal. So let's say if you have the nerve signal, the action potential was minus 50 millivolt. So a similar magnitude of chemical signal has to be generated, and that chemical signal happens through neurotransmitters. So this is a special form of a conversion of, you can say, one form of energy into the other. Neurotransmitters are released from the presynaptic axon terminal over the synaptic cleft. The synaptic cleft is the actual gap in the synapse. And these neurotransmitters are chemical signals between neurons and or a muscle. Okay. And neurotransmitters can be excitatory or inhibitory. For example, acetylcholine, it can be excitatory or excitatory. Always have a problem pronouncing this thing. It could be excitatory or inhibitory, acetylcholine. Norepinephrine tends to be excitatory most of the time. So these neurotransmitters, they have to actually be removed from the synaptic cleft. That means the gap in the synapse. Otherwise, they're going to disturb the homeostasis of the body. Let's say if you leave acetylcholine in the synapse for too long, it might actually inhibit or excite the neuron, and you might have unintended consequences. Because of this, acetylcholine is broken down by acetylcholinesterase as soon as it is transmitted into the synaptic cleft, and as soon as it has done its job, it has to be broken down. Norepinephrine, on the other hand, it is reabsorbed by the presynaptic cleft or the presynaptic neuron. And the last topic for today is the reflex arc. Reflex arc is what happens when a stimulus activates the whole neuron. And reflex arc is the route that a nerve signal follows from the sensor receptor all the way to the effectors. So first there's a stimulus, and sensory receptors pick up the signal from the stimulus, and these sensor receptors will send the information to sensory neurons, and sensory neurons will receive this information and send it to interneurons, which are called the integration centers. or in other words, this is the central nervous system. And then this information will be sent to motor neurons in terms of as a response to the stimulus. And these motor neurons are attached to the effectors through the neuromuscular junction. And these effectors tend to be muscles and glands. And then there will be a response. Let's say it's like an injection. Let's say there is an injection that's a stimulus, and when you try to take blood, that you have to prick the vein, and this signal is picked up by sensor receptors, and then it will go through sensory neurons, interneurons, motor neurons, and then it has to go to the muscle, and sometimes you might squeeze your hand because of fear. So that's the response. This is all regarding reflex arc and the nervous system. And I didn't go far from the slide, so this is going to be on your next quiz. Right. I will record another video on muscle stimulation. I don't want to do it right here. And I guess that's all for now.